Today's topic is flow cytometry and one of its variant which is based on fluorescence. So flow cytometry is a laser based technique that analyzes physical and chemical traits of cells like size, shape, internal complexity and protein expression. Fluorescence activated cell sorting is a specialized type that also physically sort those cells into different containers. It uses laser and fluorescent markers to separate and purify specific cells from a complex mixture, one cell at a time, based on the unique light scattering and fluorescence patterns. It allows rapid quantitative analysis of single cells. So, coming to the principle, it is ba- based on the principle that cells flow in a single file through laser beam. Each cell scatters light, and if fluorescently labeled cells emit light, and this scattered light and emitted light is being detected by the detector. This container has cells with different antigens over them. Corresponding to that, the fluorescent labeled antibodies are attached to them. These, these cells are suspended in the buffer. Now these cells are poured in this fluidic system, and from this fluidic system the cells comes out one at a time because of the hydrodynamic pressure built in this system. When the laser light falls on this cell, uh, the fluorescent dye absorb the light and emit the light of a higher wavelength, which is detected by the detection system. When these cells are moved from this electrical field, these cells, based on the fluorescence which is received by the detector, the cells are deflected into different containers. In the first containers, the cells which are having both red and green fluorescent antibodies are there. In this container, the cells having no antigen is there, and in this container both antigen A, A and antigen B cells are there. So main components of this system are fluidic system, from where the cell moves one by one, optical system, which includes laser and filters, detector, which has forward scatter detector, which determines the cell size, side scatter, which determines the internal complexity and differentiate the cells based on the internal complexity. Then fluorescent detector is also there, which absorb a light and emit a light of a longer wavelength, which is measured by the detector. After that, the computer system analyzes the data. So, flow cytometric analysis allows the identification of antigen positive cells. It helps in the cell sorting based on the fluorescence It also measures antigen density distribution. It measures variation in antigen expression, indicates developmental or physiological state of the cell, and same cell type may show different intensities. In multicolor flow cytometry, two color analysis is there. We, one can differentiate B and T cells. Moreover, uh, 3 to 5 color analysis is also common, and multiple antigen analyze can be analyzed simultaneously. So flow cytometry has wide applications. These can be it can be used for cell differentiation studies, for immunophenotyping, and for cell isolation for molecular studies. So advantages includes rapid and quantitative analysis, high sensitivity, multi-parametric analysis, and requires small sample. Thank you.